So the Vadose zone is the area from land surface until you hit the groundwater table. And generally when you are monitoring groundwater, you take groundwater samples and directly analyze for contaminants. But by the time you see contaminants in groundwater, the groundwater is most likely polluted. However, if you study Vadose zone, you actually know how contaminants such as nitrate are moving through the profile and making its way to the groundwater. So the way I look Vadose zone as both a time capsule. What do I mean by that? Is that it gives you an idea how past practices has impacted contaminant movement through the profile, and it can also act as a time machine. So if you can study it quantitatively, it will help you predict when your groundwater may get contaminated. So instead of being reactive by collecting groundwater samples, you are more proactive in protecting groundwater of the region. So that's why Vadose becomes very critical to study, so that you can devise a plan for a future contaminant issue that can occur. Right here, this is actually the last coring that is going on in the Upper Big Blue NRD or Upper Big Blue Natural Resource Districts. So when we go coring, we have a plan. So let's say if we are collaborating with the local NRD, they actually want to study how nitrate is throughout different locations. So for example, in this study, we are trying to collect Vadose zone cores across different phases. What are those? So NRD has 3 phases, phase 1, 2 and 3, depending on the severity of nitrate in the groundwater. If nitrate is really high above MCL, it tends to go to phase 3. So based on that, we plan that, okay, this many cores we need to collect, and which we want to do deeper cores. What do I mean by that? We want to hit the water table from the land surface. If the water table depth is really high, like this one, we have to use something like this, which is called a CME auger, because it can go much deeper than the other one that we use, which is called a geoprobe, which is much better for shallower depth. We generally take cores at 5 feet interval until we hit the water table. And as you may have seen in the video, that these are put inside a liner and capped and marked for the depth profile, which is taken to the lab, processed for nitrate and any other nitrogen in that Vadose zone sediment. Matt, at what depth are you in right now? What depth you are targeting right now?
40. This is 45.
Okay. So as I mentioned to you that we go at 5 feet intervals. So right now at this one, we are trying to reach from 45 to 50. So as you can see, the machine is pushing the course for collecting that down, which you have seen here. We have the liners inside that, which is 5 feet. And we use this rotary to really dig through and go down. So each of them are again 5 feet. So we can go deeper. And you will see as he pulls it up, we will get the sample in that end within that liner, and then they are placed together for shipping to the lab. Once we collect the cores, as I mentioned, these cores are moved to the lab. By lab, I mean Water Sciences Laboratory in the University of Nebraska, Lincoln. We take these sediments, process them together to find nitrate in these sediments. After that, we create a profile. So you have the depth and how nitrate concentrations are throughout the depth of that. Let's say in this case, if it is 80 feet, throughout the 80 feet depth. This actually gives us an idea how nitrate is moving through the profile. And if you have enough quantitative information, you can actually use modeling, such as the model that will be used here, root zone water quality model, to predict when this nitrate from the Vadose zone will make its way to the groundwater. Or if there are some management changes that is being done at the surface, how those management changes will affect nitrate in the Vadose zone before it reaches the groundwater. So basically, you can use the data set not only to understand when this contaminant is going to reach, so that you are being preemptive about ensuring proper groundwater quality. More than that, you can also devise plans that are being adopted as the waste management practices on the surface to help mitigate that nitrate within the Vadose zone before it reaches the groundwater table. So this study, as I mentioned, is the last coring that we are doing for this particular project, and we have been collecting cores for quite some time here, almost 3 years now. And once we have all the data, the plan is to use modeling to predict future contamination potentials, where I'm getting help from one of our visiting scientists, Dr. Hanuman Thalli, and Dr. Dan Snow is leading this project along with myself and Dr. Chittaranjan Ray in the Nebraska Water Center. And Vadose zone coring in the state is going for a long time. We are one of the few states who actually has such a high quality Vadose zone data, and that actually helped our state be a pioneer in managing groundwater quality, which we are highlighting through multiple research that is going on, not only in my group, but across different groups within the state and in our university.